protein synthesis is really an interesting little thing. How it all works is very complex. You can take several sessions, several semesters about micro and macrobiology, and you'd get somewhat into detail about how this all works. Let's talk first about how protein is made and how it is expressed. You'll notice obviously that your brain material isn't the same as your fingers, or your fingers aren't the same as your intestines. They all have different features, but they all house the exact same amount of information. They all have the same genes. They all have the same ways, the same programs available to them to be able to change into something else. But they are who they are. And how do we know that? Is how we're going to discuss protein synthesis and how everything works. Now, this little image I have here for you will be extremely helpful for you later on. It helps you understand how this process works. DNA, remember, is your blueprint for everything. So what happens is this. You got something saying, hey, we really, really need this in our cell. And if the nucleus is allowing us for a specific portion of DNA to show, then yeah, it's going to happen. So what happens is this little messenger RNA is going to come out, and if you remember the structure, that ribosome, the protein maker, is going to bind to that messenger RNA and create a protein. So what happens, these terms, there's transcription, here's the wordy word of saying it. You get an unraveling of your DNA, and a messenger RNA comes along and says, hey, I'm going to copy you base per base. And it does. And this process is called transcription. And all y'all know that if you're trying to transcribe word for word of something, and you've been doing it for hundreds and hundreds of pages, and you've done it over and over again, you're going to make some mistakes. So some words might get switched out and all that. That's sort of how I remember why DNA has ATGC, while mRNA uses UACG, aka it doesn't use the T, it uses the U. Transcribing can be difficult. You're writing from one language to another, you might make a mistake. However, you're using a completely new, different language. You're not using the DNA language anymore to make it. You're using the DNA language to make a protein, but if you remember from the last part, protein isn't made of nucleic acids. It's made of amino acids. So you're using a completely different language. So it translates RNA, a DNA description, to a completely new language. Just like translating English to French. Hello, bonjour. That good stuff. I have no idea if I said bonjour well at all. I probably didn't. Oh well, I'm sorry. So what happens is an mRNA is going to come along. It's going to bind. They're going to find a very specific sequence, which you'll probably get into in microbiology. It's AUG. You don't need to know AUG right now. But that's going to be the thing that this ribosome is going to look for to know where to start. As soon as it does, it's going to start recruiting these tRNA, transfer RNAs. And that transfer RNA is going to come down, bind onto the appropriate, you're going to see that it has 1, 2, 3 other nucleic acids at it that will only bind to that sequence. And if it does, and it's a match, then it's going to come on, and this ribosome is going to grab onto that amino acid and allow for the next amino acid to bind onto it. That way you form that chain. You form that primary structure. That's going back to lecture 1.5, lecture 2, last week's lecture. And that's how you form that protein sequence. So that is the formation of protein. We do need to talk about DNA and mitosis here in a little while. Let's go ahead and get into that in the next video.